about domain-specific language for chemistry. Yes, please start.
Hi all, so this talk is probably a little bit different to the style you're used to at this conference, but let me tell you about the domain-specific language I designed for peptide chemistry. So just to give you some idea into my motivation, before transitioning to my computer science PhD, I was an organic chemist working a lot in the lab, and we were doing a lot of manual work with flasks, and I had sort of inkling that we could use computer science principles and PL principles to improve and optimize our workflows, increase automation, and bring the field into the 21st century. So I don't expect you to read this word for word or understand it all, but this is a typical sort of procedure that is published in the literature, and it's quite hard to parse, it's quite ambiguous. It says things like, okay, approximately 2 mg a mil, but how approximate? Then it says the reaction mixture is stirred at room temperature. Was that room temperature in Glasgow or in Singapore when your AC breaks? So there's a problem with the literature in chemistry at the moment. There is tacit information that's taken for granted in the protocols that are written in prose, and there's some ambiguity to the language, so this has led to a reproducibility problem. Oftentimes researchers are unable to reproduce results, so there's a need to really standardize and formalize protocol descriptions. And while automation does exist, it's siloed to individual instruments typically with proprietary software, and there is a desire to consolidate sort of those workflows so you can capture them and share them. So that ties into the design philosophy of TideScript, which I developed, which is a domain-specific language for expressing peptide chemistry protocols. So peptide chemistry is a smaller subdomain of chemistry, which I'll go into more detail later. The reason for that is I wanted to hone in on a smaller subdomain first so we could properly capture the domain-specific challenges and then have scope to expand into broader territory. And I'll show how just adding fewer semantic rules can expand the expressiveness of this language, and it could maybe one day be a general-purpose chemistry language. And another philosophy is that it's designed to be hardware agnostic. So the idea is you want to have a high-level description of the experiments that are going on and have the ability to incorporate different backends to target different types of hardware architectures. Hardware architectures in this context being like laboratory instruments. And I'll show you some of the design decisions that I've made that'll enable that in the future. So unfortunately, I'm going to have to give you a quick chemistry lesson just so you can understand the design and the reasons for some of the things in this language. But I'll get to the interesting PL stuff in a moment. Just bear with me a second. So what exactly are peptides? They're just chains of amino acids. They're small proteins. They exist in biology in all living organisms. And they mediate a lot of biological processes. And the chains of amino acids are described by, each amino acid is assigned like a one-letter code. So S for serine, P for proline, etc. So when you describe a peptide chain, you describe it as a series of letters for the corresponding one-letter code for the constituent amino acids. So you might describe this as a description of a peptide, etc. Okay, and why do we care about peptides? Well, synthetic peptides are becoming increasingly more and more important in medicine. We can design peptides that will interact in a specific way with the human body for therapeutic effects, and we can also make further chemical modifications to them to enhance their functionality. And you may have heard of Ozempic, for example, is a peptide. And so the peptide chemistry essentially breaks down into two parts. You have the assembly of the chain itself, and then you have those sorts of modifications that you want to make, which are, so the peptide chain assembly itself is a process that's quite amenable to automation, is automated already, but again is siloed to specific instruments. And the solution phase additional modifications are still done manually by hand with a lot of human labor, require a lot of trial and error and repetition to optimize the experimental protocols. So you don't need to get bogged down with these chemical structures of A, a generic amino acid, B, an example of one, lysine, and C is a lysine building block. But the thing you need to take into remember is I've circled the different sort of functional groups that can react with each other. So if you imagine if you want to assemble a peptide chain one by one in a specific orientation, you need to prevent certain functional groups from reacting with each other. So you put on what are called protecting groups. That's what the FMOC and BOC is. So I'll give you a high-level diagram of this now. So you'll be assembling your peptide chain attached to a solid resin, which allows you to have the growing chain there. And you have a protecting group on the side chain and on the end as well. So what you do is, first of all, you take one of those protecting groups off, and you attach the next one on, and you'll keep going until you have the full peptide chain, and then you can remove that as long with the protecting groups. Okay, and then additionally, you'll carry out the sort of solution phase modifications that I described. So you'll take the peptide that you've made and you'll play around with it and you'll add stuff to it, you'll attach a drug molecule to it. And that was the procedure I showed you at the beginning, where it'd be in a solution, and you'll mix things together and react them and hopefully get your desired product with the desired functionality. So these are the sort of laboratory hardware that we can target. So we have batch synthesizers, these are automated synthesizers for the assembly stage, and they can produce larger amounts. We also have high throughput synthesizers as well. So these can do up to 200 at a time. And at the moment, programming these by hand is quite meticulous and error prone. So this is really where sort of PL concepts and typing and properly formalized semantics can really aid in that. And here at the University of Glasgow as well, they developed these, they're called computers, where they've got a system of flasks, reaction pumps and vessels where they can pump fluids around and sort them different things. And they can use these to automate the kind of chemical experiments that would usually be done by hand. And they have their own language that used to target this called KDL, which in the paper, if you want to read, I compare my language with theirs in more detail. And there are also these digital microfluidic boards, which are really cool. They just consist of a plain array of electrodes, and you can sequentially activate the electrodes to manipulate discrete droplets of fluid. And you can move these around and use this to automate small-scale experiments, analyses as well, and they've seen some application in automated analyses like PCR tests, etc. Okay, so now onto the PL stuff. We have this protocol language that I designed. So we take an imperative approach where there's a series of statements that we want to execute one by one, and the statements fall into two categories. The declaration statements, where you want to declare the materials that are present within an experiment, like this particular molecule, this particular solution exists, and we're going to play with it. And any operation statements, where you actuate the actual components of the hardware architecture, whether that be dispensing or heating, etc. So to go into more detail about declaration statements, so you might have a particular solvent that's involved, as DMSO. So you declare that, you say DMSO is a solvent that I'm using, and it pulls from a table that has salient information about the solvent, its density and polarity, etc., which will be relevant for downstream processing or perhaps typing later on. And then you declare like a molecule that you might be using, and there's two forms. So this here is just the formula description, and when you pass in the formula, the compiler can infer the molecular weight of the molecule, which will be relevant later. And the second one, molecule B, what they're passing in there is called the smile string. Now, the smile string has its own set of rules. You can check the grammar for smile strings on opensmiles.org, where this describes the chemical structure as well. It's used a lot in chem informatics and computational chemistry, and this can give even more information that will be useful in downstream analyses. If we know what the structure we're expecting, we can relate that to analytical results that we get. Peptide, you can pass in a description of the sequence again, and again, the compiler can infer information like the molecular weight, and for linear peptide, it's smile string and structured as well. And then, finally, you say, I have a solution X, and it is this molecule dissolved at this particular concentration in this solvent. The salient difference between the solutions when they're instantiated exist, and the compiler assumes it to have a location. It's a real physical thing that exists. But the peptides and molecules are not, they're abstract concepts. You say that this is the molecule we have, but then when you instantiate the solution, that has a container, and it's a physical entity that has the previously described things dissolved in it. And then we have operation statements. These are statements that will actually actuate the components of the system architecture itself. So you'll have an effect on the sort of state of the program, and it might be changing the temperature, heating or cooling, or you can even instantiate new solutions by mixing previously declared ones. And we have the keyword agitate here, and this is part of the hardware agnostic design. So agitation can take different forms depending on which kind of hardware you're using. It could be shaking, it could be stirring, inverting, etc. And then another concept we have are protocols, which are just functions essentially. You have a series of statements in the body of the protocol, and then you parameterize those, and you can pass in arguments to the parameters. So you can define a protocol like this here, where it's shake solution, and then you can pass in a previously declared solution, etc. So with all this in mind, we can look back at the original procedure that I showed you at the beginning and see how we can express this in TideScript, okay? So you can declare the peptide, pass it in, the molecule that you're using, and the solvent, and then you're going to declare two solutions here. You have a solution with your peptide dissolved in, and your solution with the molecule that you're attaching on dissolved in as well. And you can see here you're describing the concentration of them as well, and there's a superficial support for units here. See, this is described in millimoles, and this is described in mgs per mil. Now, supporting units is less trivial than you'd think, actually. So at the moment, it's just a syntactic sugar. So concentration is always calculated as mgs per mil, but as the compiler powers down, it calculates and infers what the concentration should be. So then you define a protocol, and you would have this is a protocol that returns a solution because you mix solutions that you pass in, and you can change the temperature, agitate, etc. And instead of saying we add 5 mils to this or 1 mil of that, we have equivalents. So we don't get, that's a chemical term, but you don't get too bogged down. Essentially, a one-to-one equivalence means like a one-to-one amount of molecules. So with the equivalence semantic rules in there, we can call the protocol and call it at different scales as well, again with the unit support. And I can go over just some of the formal specification. There's a lot of syntactic rules here, but again, as I say, there's a syntactic, at least, support for units. So if we consider the semantics, if you have a program P and the state of the program S, the state of the program essentially involves the peptides, molecules, solvents, solutions, and the protocols you've defined. And the operation statements you make changes to that state. So, for example, here are some of the semantic rules. I'm not going to go through every single one by one, but if you want to declare a peptide needs to not already exist in the program state already, and same with the molecules and solvents. And then the solution one is slightly different. It's where when you declare a peptide or a molecule that is dissolving in a solution, it needs to already previously been declared, and then you'll append that to the state of the program at a particular concentration. Again, with the protocol declaration, we have a series of statements and a series of parameters. and when you call that, you'll pass in arguments. So these are the operation statements as well. So again, for example, when you dispense a solution, you instantiate a new solution of a volume n that you've described, and the old solution will have its volume decreased by n. Just an example of the sort of semantic rules we have that describe the domain that's going on here. And when we call protocol, you essentially just execute on a series of statements and swap the parameters for the arguments, is what's that trying to say. Okay, so all of this, an example I show you, expresses the in-solution phase modifications that you make to peptides. And then you can add, I can show you how we can add a few more semantic rules. So we can express the solid phase, the peptide chain assembly that I was showing you at the beginning. So we just need a few additional concepts for resins and reaction vessels, and a few additional operation statements. And when you consider, for each amino acid, there could be a dozen or so steps to couple it on. So when you have longer chains, you're talking of hundreds of steps. And using both protocols and repetition statements, repetition is just a for loop, essentially, just to repeat the statement a number of times. We can express quite a large number of steps in a small number of lines of code and show how this language can actually be quite expressive. So again, we have superficial syntactic support for units and additional operation statements to allow us to express what we're talking about here, and repetition as well. How am I doing for time? Okay, and then again, there's additional semantic rules, the repetition, the agitations, and the ability to add and drain of these. I didn't include all of the rules here, but if you're interested, you can see it in the paper. So again, I don't want you to read this word for word. This describes, on the left, it describes the series of steps to remove a protecting group, and on the right, it describes the series of steps to couple the next amino acid on. And it's all quite wordy, but we can express it using the protocols here, and you can see it's pretty clear what's going on. We have our reaction vessel, and we have we can define a wash cycle for that, and a shake cycle for that, and then we can take the deprotection that was on the left and the coupling that was on the right and define protocols here. I can express that procedure that way, and then finally we can add them together in one cycle, and then we can iterate through a sequence. So we've defined the coupling cycle for one amino acid, and then we can pass in a sequence and iterate through it. So what we've expressed here is actually hundreds of steps, and this is the kind of thing that will be executed on the batch synthesizers that I showed you earlier, or the high throughput ones. Okay, so why bother with all this? Well, I could have just taken an approach where it was just an operational approach where you just describe the movement of fluids from A to B, but instead there's some encoding of a modeling of the underlying chemistry that's going on. And the reason for that is it enables for future work to really take advantage of PL concepts that could really improve and optimize our workflows in future. So, as I say, we're not able to express every single protocol yet that exists in literature. Currently working on a survey of what we can and cannot express, but there's currently an escape hatch built in where you user might have to do something manually. But as I've shown, we can add a few more semantic rules and really expand the type of things that it's able to express. And in terms of future work then, so yeah, as I described, there's a reason why we're properly describing the materials and molecules, etc., that are involved, because we want to categorize molecules and reactions. When you think if a chemist is carrying out a particular reaction, they might be trying to achieve a specific goal. It could be a deprotection, a bond formation, conjugation, etc. But to achieve the desired outcome, there might be certain conditions that they need to meet, and a sort of type system could catch these errors early. So if you have the structure of a molecule here, we can abstract away the sort of different functional groups. And sort of try and predict how they're going to interact and have early stage optimization and prevent errors that occur. The type of errors that we could make, particularly when I mentioned this high throughput synthesizers that have 200 plus peptides at once, there's many errors that are often made, and there's an acceptable iteration rate in where they'll make 200 or so peptides, and then maybe 50 or 40 of them won't work, but they just accept that and move on. But instead, we could improve the efficacy of these therapeutic cocktails by catching that early and making sure that these are the kind of mistakes they can make and they can be prevented from making. So, for example, typically when you want to carry out a coupling cycle, you'll probably want at least 5 equivalents of a thing. You don't want to perform a capping reaction at the wrong stage, because that can kill your synthesis. Low-hanging fruit as well is you try and dispense 80 mils, but you don't actually have that in your thing. So that's the kind of domain-specific typing that I want to incorporate in the future to improve those workflows. And another elephant in the room is that chemistry is a stochastic process. When I used to say, currently semantically, we say change the temperature of the solution to 21 degrees, and it's magically 21 degrees, but that's not the case. It's much less deterministic than that. It may take time to heat up. You might need to incorporate some dynamic feedback in the runtime to make sure it achieves the temperature you want. And in terms of the reactions themselves, they might not go the way you expect, the way you want. And any typical flask, there's order of 10 to the 23 molecules in there, and there's a lot of side reactions, a lot of things that are going on. So you want to have a probabilism in there to account for that and work in some probabilistic semantics in the future. So in conclusion, I've designed TideScript as a protocol language to express peptide protocols unambiguously with scope for future execution on actual hardware. It's working on at the moment. The particular design decisions that I've made for the scope of future work, and in the future we aim to develop a richer type of system and incorporate probabilistic semantics. So that's me.
If you have a question for more Q on the mic.
I enjoyed the chemistry lesson. And I was thinking, when you mentioned the agitate statement, and you said it was like abstract over different hardware. At the end, you mentioned that there are escape hatches, because that's what I wondered when I saw the agitate statement. You said it abstracted over different ways of agitating in specific machines. And I was just, it reminded me of, I don't know, as in blocks with machines.
So it reminded me of ASM blocks in C, where you can kind of bypass, that's like an escape hatch through the compiler. So if you really, really want to express something in assembly, then you can do that. And I was wondering if maybe chemists, I don't have background in this, which is why I enjoyed the lesson, but I was wondering if chemists might want to be able to go through that and to say, no, actually my hardware really does need this specific mechanism, so you need to provide hinting or something of like extra statements on the agitate statement or something like that.
Yeah, exactly. So going through like some of literature procedures, sometimes they involve taking and adding it to a tube and centrifuging it, and the movement from there is non-trivial. There's no automated hardware where we can do that. So yeah, so there are escape hatches analogous to unsafe perform IO, where you, okay, I'm going to intervene and carry out a manual performance, because that's the only way that some of these can be expressed and actually executed in the future.
Thank you.
So I actually don't know much about protein synthesis, but I was wondering first, is it mostly a manual procedure now? And is that why you're trying to come up with a language that would make it easier to automate? And also, why did you choose this as your domain? And would you want to expand it to other I don't know, applications in chemistry or biology, because I can see it being useful in a lot of different experimental procedures that would work similarly.
Yeah, well, the honest answer is I already knew about peptides, so it was easy. But yes, absolutely want to expand into broader domains. That's definitely the design philosophy from the beginning. And to answer your first question, truth be told, the peptide chain assembly is an automated process largely, but there's still a lot of suboptimal procedures that go on and a lot of cost to academics and industries when mistakes are made that could have been prevented. And there's all kinds of like pre-procedure optimizations that you can incorporate that I'm hoping a strong type system can aid in, especially when you consider the high throughput scenarios where there's hundreds going through at a time and there's just an acceptable rate of iteration.
Yeah, I think that's pretty cool.
Okay, thank you.
Hi there. Really neat work. Really enjoyed your presentation. So right now it sounds like this is protocol specification. You're not running it on hardware yet, which makes a lot of sense as a place to start. But I was curious if you've run this by any other chemists and like what the reaction of scientists to it has been, if you've had a chance to do that yet. As somebody who works a lot with physicists, I'm very curious.
No, I've not done yet, but there are plans at my university to do a talk to the chemistry department. The only chemist I've run it by is myself, but yeah, I definitely plan to do that. That's very useful because there's certainly things I could have missed.
Great, yeah, excellent. Thank you so much.
Thank you.
Hello, thank you, nice talk. What do you think -03 optimizations would look like for your system? I mean, can you imagine, I don't know, constant propagation in your system, say, or something like, I don't know, redundant code elimination?
Yeah, well, that's the dream. The supervisor was talking about this. Perhaps we could reorder steps in ways that we couldn't have previously imagined. That's quite a hard problem, and there's a lot of machine learning efforts into this sort of thing, and it's an ongoing research area, but it's
very exciting. Thanks.
So often scientists have a whole bunch of like other tools they use for kind of analyzing, like taking in data from experiments and then generating various kinds of visualizations. There's usually like domain-specific things, particular kinds of tasks. So have you thought about how you could kind of connect this with that kind of, with those other kinds of tools, like, you know, for preparing papers or visualizations? Because it's sort of like you've got something about the methodology which kind of implies something about the kind of results that are going to come out, like the shape of them.
Well, so the integration here is like the reason you pass in like chemical information rather than just using strings or what have you is if you take an analysis further down the line, if the compiler or the system you have knows what chemical structure to expect, then it can automatically carry out analyses on the sort of analytical data that's collected from these experiments, things like NMR and HPLC. It's beyond the scope of this talk, but yeah, that's why those decisions were made in terms of the declaration statements.
Yeah. So maybe there's a chance to like connect it with other tools in the
Yes.
future, right? I mean, there's a huge number of NMR tools out there, for example. Yeah.
Yeah, absolutely.
Yeah.
That would be the thing.
Yeah, thank you for a very interesting talk. I understood that when you want to extend your experimental process, you have to extend your protocol language. So from the prospective language design, programming language design, extending a language itself to apply that language to a slightly different application domain is not a good idea. Usually, that kind of extension should be implemented should be implemented as a library. So do you have any idea to support that kind of generosity, to add some sort of generosity to your protocol language?
Yeah, so perhaps top-down design is better than the bottom-up one. Well, however, I do hope that, I mean, there's already a description of solutions to molecules which can describe a lot of chemistry that's not peptide-specific already. So I'm hoping that like a small expansion of the semantic rules will be able to capture general chemistry, and then people will be able to have smaller libraries for smaller subdomains. the peptide chemistry was chosen as a sort of proof of concept initially.
Thank you.
Thank you.